Hello, my name is Zachary Fossler, and today I'm presenting a literature topic on improving nitrogen agronomics in fertilizers by use of urea-hydroxyapatite nanoparticles. To begin, I would like to talk about the Haber-Bosch process. In 1913, two German chemists, Fritz Haber and Karl Bosch, came up with the Haber-Bosch process, and it is shown here in the diagram to the right. As you can see, atmospheric nitrogen is combined with compressed hydrogen gas at a 1:3 volume ratio, respectively. The mixture is passed over a catalyst, which is usually iron, at a temperature of 400 to 450 degrees Celsius, and at an extreme pressure of around 200 atmospheres. Now, this is to produce ammonia, which is a key source of nitrogen in fertilizers. This reaction is considered to be one of the most important chemical reactions discovered, and it has contributed to the Green Revolution, which is responsible for the increased food production that has sustained the expansion of the human population from around 2 billion people in the early 20th century to what it is today of around 8 billion people. Now, the problem with conventional fertilizers is that they are very inefficient, with around 50 to 70% of the nitrogen being lost to the soil and not assimilated by the plant. Nitrogen runoff and leaching can have devastating effects on the environment, such as eutrophication of lakes and other bodies of water, and can lead to groundwater contamination. Also, nitrogen is one of the most expensive nutrients to supply in plant production, with an annual usage of around 85 to 90 million metric tons used worldwide. However, only around 45 million metric tons is actually uptaken by the plants. Therefore, there is a great need to increase the nitrogen use efficiency in fertilizers. So the goal of this experiment was to develop an environmentally benign nanohybrid that is coated with the nitrogen-rich molecule urea. It must be easily synthesized with high loading efficiency, and to do this, the nanoparticle must have a high surface-to-volume ratio. It must also improve the nitrogen agronomic efficiency by slowly releasing nitrogen to the plants over time. Now, the mineral hydroxyapatite was chosen for this experiment, and that's because it has been extensively studied in the fields of material science, biology, and medicine. It is a key constituent of human and animal hard tissue and is commonly used as a bioceramic due to its biocompatibility. Also, rock phosphate, which is a form of hydroxyapatite, is already used in fertilizers as a source for phosphorus. And also, the reactive surface functional groups allow for the surface of the nanoparticle to be modified. Now, the synthesis of the nanoparticle was performed through a one-step in situ approach, where solid urea was dissolved in a solution of calcium hydroxide and mixed for 45 minutes. Phosphoric acid was then added dropwise to the solution, and it was stirred for another 2 hours. After that, the solution was then flash dried at 60 degrees Celsius, and the optimum ratio for urea to hydroxyapatite was found to be at a 6:1 respectively. If any higher ratio of urea was added, their phase separation began to occur. Now, this slide shows the morphology of the urea hydroxyapatite nanoparticles, and in figure 3a shown here is a TEM image of the hydroxyapatite nanoparticles, and what it reveals is they have a rod-like shape with diameters between 15 to 20 nanometers and lengths ranging from 100 to 200 nanometers. The high-resolution TEM image shown here in figure 3b shows two distinct phases, the urea phase and the lattice fringes of the hydroxyapatite. The lattice are perfectly aligned with a spacing of 3.5 angstrom, which corresponds to the interplanar spacings of planes for hexagonal hydroxyapatite. Figure C just shows again the hexagonal morphology of the urea-coated hydroxyapatite nanohybrid, and figure 3D is an SEM image showing how the nanoparticles have a bead-like structure after they are synthesized. However, after spray drying, the nanoparticles form these rod-like structures as shown here in figure E. Now, this next slide compares the electron diffraction pattern of urea and urea hydroxyapatite nanoparticles in figure 2.1, with A here being pure urea and B being the nanohybrid. It also compares the PXRD patterns of hydroxyapatite, urea hydroxyapatite nanoparticles, and pure urea in A, B, and C respectively, in figure here 2.2. Both of these figures help confirm the polycrystalline nature of the Urea-Hydroxyapatite nanohybrids, with figure 2.1b showing the rings formed by the many crystallites present in the sample, and figure 2.2 showing the broad and low intensity peaks in the PXRD patterns. The crystallite size was found to be around 18 nanometers, which is consistent with the TEM observations of the average size of the nanoparticles. Also, there was no observable shifts in the PXRD peak positions before and after the surface modification of the hydroxyapatite nanoparticles. And what this indicates is that the attachment of the urea to the nanoparticles is weak and does not substantially modify the hydroxyapatite nanoparticles. Now, the interactions between urea and hydroxyapatite nanoparticles were further studied using XPS, which is shown in figure 4, where the calcium 2P, the nitrogen 1s, and the phosphorus 2p binding energies were monitored between pure hydroxyapatite and the nanoparticles and the urea-coated hydroxyapatite nanoparticles. Now, the most significant shift in peak position towards higher bonding energy was shown here in figure 2, and it corresponds to the nitrogen 1s core level for the urea-coated hydroxyapatite nanoparticles. And this is indicative of a new bond being formed between the urea and the hydroxyapatite nanoparticles through the nitrogen atom of the urea. When looking at calcium 2p XPS spectra, there is also a noticeable shift towards a higher binding energy. This suggests that the chemical environments around the calcium 2 plus ions in hydroxyapatite is changed in the presence of urea. Also, there was a small shift in the phosphorus 2p core level spectra, which is possibly due to the forming of hydrogen bonds between the phosphorus groups of the hydroxyapatite nanoparticles and urea. However, the XPS data indicates that the main bonding mode is through the metal to ligand interactions between the nitrogen atoms of urea and the calcium atoms of the hydroxyapatite. FTIR helps confirm that the hydrogen bonding modes are present. When comparing the amine stretch of pure urea to that of the urea-coated nanoparticles in figure 5, you can see there is a shift, which means the nitrogen and hydrogen bonding environments are altered. The shifts in the carbonyl and the nitrogen-carbon-nitrogen bond of the urea region also help to confirm that the bonding of urea to hydroxyapatite occurs through the nitrogen-hydrogen environments. Next, in order to understand the surface characteristics of the hydroxyapatite nanoparticles once they were coated with urea, BET analysis was carried out. Table 1 here shows the results of pure hydroxyapatite. It had a surface area of 81 meters squared per gram, and according to the BJH method had an average particle volume of 0.28 centimeters cubed per gram, with an average particle diameter of 14 nanometers. Comparing this with the urea-coated hydroxyapatite nanoparticles in a 1-to-1 ratio shown here in Table 2, it shows that the surface area decreases to 56 meters squared per gram, as well as the average particle volume to 0.25 centimeters cubed per gram, and the average diameter actually increased to 17 nanometers. The reduction in surface area indicates successful coating of urea to the nanoparticles, and this was further expressed once a 6-to-1 ratio of urea to hydroxyapatite was analyzed, where the surface area was calculated to be 1.8 meters squared per gram, thus confirming a greater coverage of urea on the hydroxyapatite nanoparticles, and this is in agreement with the TM images shown previously. Now, to understand the nitrogen release behavior of the urea-coated hydroxyapatite nanoparticles, a release water test was used, and the setup is shown here in figure 6a, right here. A sample of either pure urea or the urea-hydroxyapatite nanohybrid was placed in the chamber shown here, with each sample containing 0.92 grams of nitrogen. Distilled water was then pumped through this chamber at a rate of 3.75 milliliters per minute. Elutes were then collected every 20 seconds and analyzed using FTIR. The data obtained is shown here in figure 6b, where 99% of the nitrogen in pure urea was released in the first 320 seconds, as opposed to 86% release of the nitrogen in the urea-coated hydroxyapatite nanoparticles within 3,820 seconds. This showed that the release rate was reduced by approximately 12 times the amount of pure urea. Now, the release kinetics were studied using pre-existing models, and according to the R-squared value, the Higuchi model gave the best fitting line, shown here, and it follows a Fickian diffusion model. Since it follows this form of fixed diffusion law, it can be assumed that the release mechanism is analogous to the release of water-soluble molecules from a homogeneous matrix through diffusion, which is commonly found in slow-releasing drug formulations. So once the nanohybrids come into contact with the soil, the urea releases from the hybrid in a slow and controlled manner through a diffusion control mechanism, and leaves the nanoparticle in the soil, and these nanoparticles are not absorbed by the plant. The bioavailability of this slow-release urea-hydroxyapatite nanohybrid was evaluated at the Rice Research and Development Institute in Sri Lanka, with rice as the model crop. The field testing was then extended to certified farms. The field trials revealed that a savings of up to 50% in urea consumption was attained by using the urea-hydroxyapatite nanoparticles. Figure 8 here to the right compares the yield per hectare of rice with no fertilizer, which is T1, the recommended amount of granular urea, which is 100 kilograms of nitrogen per hectare, which is here in T2, and the urea-hydroxyapatite nanohybrids at 50% the recommended amount of nitrogen, or 50 kilograms per hectare, which is here in T3. What the data shows is that the nanohybrids actually outperform the pure urea, even at half the amount of urea. Additionally, the nitrogen, phosphorus, and potassium contents of the leaves obtained from the rice paddy plants 4 weeks after germination were compared for the plants with the recommended amount of urea and the plants with 50% of the recommended amount of urea. And this was added by the urea-coated nanohybrids right here. The results obtained are shown in Table 3. The table shows that the amount of nitrogen uptaken by the plant is maintained in the urea-coated hydroxyapatite nanoparticles, even at half the recommended amount of nitrogen. So to conclude, the experiment showed that an environmentally benign urea-coated hydroxyapatite nanoparticle was successfully synthesized. It was revealed that the urea interacted with the hydroxyapatite nanoparticles through its amine and carbonyl groups to produce a nanohybrid that exhibited a slow release of nitrogen to produce a better rice crop yield at half the recommended amount of nitrogen. Here are my references, and I would like to thank Dr. Santra, Dr. Kiava, and Dr. Kolpashchikov. Thank you.